Hello, this is Taranum Khan from the Department of Microbiology, KG Somaya College of Science and Commerce. The topic for today's presentation is transport of protein into the nucleus. As we know, within a cell, there are some proteins which are synthesized in the cytosol and some proteins are synthesized in the nucleus. So the proteins which are synthesized in the cytosol, like enzymes, histones, transcription factors, all these proteins needs to be transported inside the nucleus. So this transport occurs through channels on the channels or pores on the nuclear membrane surface known as nuclear pore complex. For example, if this is the membrane of the nucleus, there are nuclear pore complexes NPC which allow the transport of protein inside the nucleus. Now let us see how this transport takes place. So one of the important factors which help the protein direct inside the nucleus is known as nuclear localization signal. This signal will guide the protein that it has to go inside the nucleus and not anywhere else, like not in mitochondria or nor in a Golgi body. So nuclear localization signal ensures that the protein Nuclear localization signal ensures that the protein is going inside the nucleus and not anywhere else. Okay, so if this is my protein of interest, there will be a nuclear localization signal present on that protein. This nuclear localization signal will import the protein inside the nucleus. Now the location of this signal may vary. It is not necessary that it will present at the terminal end only. It can be present anywhere in the protein, but the main aim of the NLS is to transport the protein inside the nucleus. So how is this present? The characteristic of nuclear localization signal is they are nothing but short stretch of amino acids, typically rich in positively charged residues like lysine and arginine. They are almost located anywhere. They form loops or patches on the protein surface. They are present almost anywhere, forming loops or patches on the protein surface. Now, there is a protein known as RAN protein. RAN is a molecular switch which ensures the directionality of the nuclear transport. It will ensure and take care that the protein which are destined to go inside the nucleus will go inside only, and one which are destined to come outside the nucleus will come outside. Now let us see how RAN is maintaining the directionality of the transport. As I said, RAN is a molecular switch. RAN exists in two forms, RAN GTP and RAN GDP. RAN GTP is the active form of RAN. Means whenever the RAN protein is bound to GTP, it will be in active form. And RAN GDP is the inactive form. So when RAN is bound to GDP, it will be in inactive form. Now, there is an enzyme known as you all can see here, RAN GTPase. RAN GTPase has the capability of converting RAN into active and inactive form. Now, as you all can see here, the predominant form found inside the nucleus is RAN GTP. RAN GTP is created by the action of RAN GEF. What is RAN GEF? It is guanine, RAN, guanine exchange factor. RAN guanine exchange factor. So what is the function of RAN GEF? RAN GEF will convert RAN GDP to RAN GTP. Okay. So RAN GEF is RAN guanine exchange factor, means it will convert the inactive form of RAN, that is RAN GDP, to RAN GDP. Similarly, the same process is going on in cytosol, as you all can see that in cytosol the predominant form is RAN GDP. So there is an enzyme present in the cytosol known as RAN gap. Gap stands for guanine RAN gap. The function of RAN gap is converting RAN GTP to RAN GDP. So it is guanine activating factor. So it will activate the GTPase activity of RAN and convert RAN GTP into RAN GDP. So this is how the distribution of RAN is maintained. RAN GEF ensures that RAN GTP is present in the nucleus, and RAN gap ensures that RAN GDP is present in the cytosol in predominant form. Okay. So now let us move ahead and see the GTPase cycle. So as discussed, in the cytosol, RAN gap, that is GTPase activating protein, promotes GTP hydrolysis. So what it is going to do? It will convert RAN GTP to GDP. This is how RAN GDP is present in more amount in the cytosol because of the activity of RAN gap, which is GTPase activating protein. Inside the nucleus, as we discussed earlier also, inside the nucleus, we have RAN GEF, which is guanine exchange factor. It will convert RAN GDP2 into its active form, that is RAN GDP. This ensures the directionality of the nuclear transport. The spatial distribution of RAN ensures the directionality of the nuclear transport. Now let us see how the import of the protein is actually taking place with the help of these proteins. Nuclear import. Now, one of the major role played by nuclear import is by nuclear import receptor. So there is a receptor. This is nuclear import receptor. On the receptor, the protein of interest to be transported will bind. So nuclear import receptors are soluble cytosolic proteins. Okay. So nuclear import receptor will bind to the protein of interest and carry this protein of interest inside the nucleus. Now let us see the entire import cycle. How is the import taking place? So the step one is, once the protein is synthesized in the cytoplasm, if the protein is destined to come inside the nucleus, it will have nuclear localization signal. Protein containing nuclear localization signal bind to importin. Now, what is importin? Nuclear, another term, it is another terminology used for nuclear import receptor. So, protein containing nuclear localization signal binds to nuclear import receptor. Now, once the protein is bound to the nuclear import receptor, it will travel inside the nucleus through nuclear pore complex. So there are nuclear pore complexes present. This nuclear complex is rich in Hg repeats. that is phenylalanine and glycine. So this receptor containing the protein of interest will bind to the FG repeat on the nuclear core complexes and the proteins then travels inside. Once the protein is inside the nucleus, now suppose this is the receptor containing the protein of interest. Once this is inside the nucleus, what is going to happen here is the GTP, GTP present inside the nucleus will bind to the import receptor, leading to release of this protein. So the protein will be released inside the nucleus. Protein will be released inside the nucleus and the empty receptor is then recycled outside the nucleus so that the next cycle can begin. So let us quickly go through this cycle one more time, that what is happening in the import cycle. The protein which is synthesized in the cytosol, it contains the nuclear localization signal and it will bind to nuclear import receptor. Once the protein of interest binds to nuclear import receptor, then this import receptor travels through the nuclear core complex. Now, on what it is binding? It is binding to the FG repeat present on the nuclear core complex. This is how the receptor and the protein of interest will come inside the nucleus. Once it is inside the nucleus, we have already discussed that inside the nucleus, the concentration of RAN GTP is in high amount. So this RAN GTP will bind to the nuclear import receptor, leading to the release of the protein of interest inside the nucleus. Now, once the protein is released, the receptor containing the RAN GTP is transported outside the nucleus, that is into the cytosol. Once the receptor and RAN GTP are there in the cytosol, we all know what is present in high concentration in the cytosol? RAN gap. This RAN gap will convert the RAN GTP to RAN GDP, releasing it from the receptor, and the empty receptor is now ready to begin the next cycle and transport some other protein. This is how the import of the proteins takes place inside the nucleus. Now the next is nuclear export. In a similar mechanism, the mRNAs and other proteins which are formed inside the nucleus, they needed to be transported outside the nucleus, that is into the cytosol. As we all know, transcription takes place inside the nucleus. But once the mRNA is formed, the process of translation will take place in the cytosol. Hence, it is important that this mRNA needs to be transported outside. Now, as in a similar mechanism, how the protein which are supposed to be transported inside the nucleus contains nuclear localization signal. The proteins which are destined to be transported outside the nucleus contains nuclear export signal, which is NES, nuclear export signal. Now, as we have nuclear import receptor, in a similar manner, we have nuclear export receptor. So now the mechanism is same. The protein of interest will bind to nuclear export receptor, and then this protein is taken outside into the cytosol. Let us see the process step by step. So now let us see the complete export pathway. So earlier also step 1 will be binding of the protein of interest to nuclear export receptor. The receptor carrying the protein of interest will bind to the FG repeats on the nuclear core complex. Nuclear export receptor, as mentioned here, are known as exportins. So this exportin plus the cargo, that is the protein of interest, along with RAN GTP, as we know RAN GTP is present in large amount inside the nucleus, will bind and travel through the nuclear core complex. Once into the cytoplasm, the RAN GTP hydrolysis takes place. As we know, in the cytoplasm, RAN gap concentration is present in large amount. So the hydrolysis of RAN GTP will take place inside the cytosol. The protein of interest will be released and the empty receptor will be recycled back again inside the nucleus. Once the empty receptor is inside the nucleus, the RAN GDP is converted back to RAN GTP by the RAN GEF, which is present in large amount inside the nucleus. So now let us summarize the entire mechanism of whatever we did today. So let us discuss the important concepts. So the proteins which are destined to be inside the nucleus contains NLS, that is nuclear localization signal, and the proteins which are destined to go outside the nucleus contains NES, which is nuclear export signal. The receptors which target protein inside the nucleus are known as importance, that is nuclear import receptor, and the receptor which takes proteins outside the nucleus are known as exportin, which is nuclear export receptor. What is maintaining the directionality of the transport? The distribution of the RAN protein. RAN GDP is present in large amount in the cytosol and RAN GTP is present in large amount in the nucleus. What is the gateway of the transport? Nuclear pore complex is the gateway. Both the import and the export receptor binds to FG repeat present on the nuclear pore complex. RAN GPP's activities maintaining the directionality. While the import and the export machinery are mechanically similar to each other, but they function in opposite direction. Import receptor will bring protein inside the nucleus and export receptor will take the protein outside the nucleus. Thank you.